I would like to go from sugar to photosynthesis, the process before, to get the sugars. In this sense, it's not out of control. And thank you for who created all these nice session names. It made me think a little bit, what is out of control? Are we out of control measuring photosynthesis with that white device on all those our mobile devices moving through the field? So I tried to make a little video as a starting point, putting all those mobile devices, all those mobile phenotypes that we have in Australia and Europe, and put them together. So there's a mouse. Let me try to start the video. And okay. Well, the videos don't work here. Sorry about that. But I think it illustrates very well. We are out of control sometimes. And it makes us, we have to think about it. Are we really out of control with all these phenomobiles, all these techniques being thrown upon us in field phenotyping? Luckily, I also back to earth, back to the newspapers, yesterday's newspapers in the Netherlands, high-tech, in the field for the farmers. Here, a farmer's son, 20 years old, he puts his drone into the field and measuring over all the fields and measuring the colors and the different parts, says here you have to put more nitrogen, there you have to put less. And the farmers, they said, no, what I want to see what you cannot see. I want to look below ground. And that's what we have for. So he said, okay. So he was not completely there yet, that farmer's son. He couldn't convince the farmers completely. And he also had some problems in giving the data to the farmers. He said, okay, I have my data here, several gigabytes of images, and he wanted to provide them, but he couldn't give it because there's not a fast internet. And I think that covers and illustrates pretty well also the conference of the day. Tomorrow we have the session about data. Getting the data to the users still sometimes is a problem. But the other part is also having the farmers on the right track, or having the breeders, or having all of them on the right track. And I think we're pretty much there. And there I go back to a slide some of you may have seen earlier, the basis of field phenotyping. And I would like to go through these 5 pillars of field phenotyping, maybe also as a perspective of a farmer. If you go from the right side and say like environmental sensors, well, the weather is really nice. I will plant a week later because it's raining today. Can plant in the experiments. I can plant my fields on the southern slope 1 week later because the crop cycle will be much faster. It's raining. I can use my tractor with double wheels. It's a better positioning system. And the sensors are the eyes all of the times. And then in the end, it's the yield. But the yield, there's also a hidden part. Resilience, we are dependent on other traits that we cannot see, as we said, not the yield in the end. And that's the 5 pillars of phenotyping, a little bit the other view to it, but also in field phenotyping, these 5 pillars, environmental sensors, always determine your environment. Experiments is what they all do together. Having a good positioning system is key to position your sensors and to measure all the traits. Today I would like to focus on only one sensor, and in this case I would like to focus on the sensor that we usually cannot see, and that is measuring photosynthesis. Measuring, in this case, fluorescence. Most of you may have also experience with gas exchange. I do skip that because, well, it's a nice measure of the end of the CO2 fixation in the photosynthesis process, but it is a little bit time consuming to get that, and in a little bit throughput that may give some difficulties. When I talk about fluorescence, it's the reflected part. I mean, the light is absorbed by the leaf. That shows basically this basic picture of that light absorbed and leaf. 80% of the absorbed light goes into photosynthesis, or I must say can go into photosynthesis. Another part goes to fluorescence. Measuring this fluorescence we can do with different sensors, and we can have an idea of efficiency and photosynthetic capacities. This fluorescent signal here expressed in the wavelengths, you see it here as a big red blob at 2 wavelengths, mostly the 680 and 740, that we can measure with spectrometers, but we can also measure that with active fluorescence approaches. Active fluorescence approaches, on the left side, the pump fluorescence, you give a saturating pulse and you receive back your fluorescence. And this method of Waltz in the 20s is much used, and a lot of people have used it over the times. And it also made the measurement of fluorescence widely accepted. It's not that we all know it very well, but it has paved the road for doing photosynthesis measurement by fluorescence. Now, recently, we have the more abstract approach, if I may say. There's sunlight, comes on the leaf, it's absorbed, and plants fluorescence. By tuning our spectrometers very finely and measuring in the Fraunhofer bands the reflected or the fluorescence, we can measure also sun-induced fluorescence. We developed for that a sensor in an airplane, and because of the development, we're moving also to satellites. But these are much larger scales. We're going there for the small scale clip-on, and the other one, the scale at satellites, these are several meters or up to 300-meter areas. That's not really what I think about high-throughput phenotyping. There you have to be right in the middle, where we look at proximal sensing, can be a meter, we can put it on our phenomobiles, but also tractors or airplanes maybe in the future. And I would like to go through two proximal sensing approaches, one active and a few slides on a passive approach. First with the passive approach. Here you see a happy Laura, colleague of mine, measuring with the so-called Floxbox in a lattice field. This Floxbox is a very temperature-controlled spectrometer that can measure sun-induced fluorescence. However, I still think it's rather slow. It takes 1 minute to acquire these data, and it's valuable to obtain a lot of knowledge, but I will not go much further into this in this example for high-throughput phenotyping. But it's certainly interesting. And the sun-induced fluorescence approach is also interesting, and although it may be a little bit slow at the moment, still for our understanding it's valuable. Here you give an example of a bigger sensor on the left upper side, also measuring sun-induced fluorescence in combination with structure. Francisco Pinto, a former PhD student of us, now working at CIMMET, he nicely combined the signal of sun-induced fluorescence with the structure of the canopies. And obviously, if we measure photosynthesis, also in the next slide that I will show you, and I say this is canopy photosynthesis, it's the integral of the function of structure and photosynthesis at leaf level. Keep that in mind, although I will only refer to it as photosynthesis. But still, this is very nice for a deep understanding, you may call it deep phenotyping, of fluorescence as a photosynthesis trait to measure. But the rest of the talk I would like to spend a bit more on this sensor. It's a light-induced fluorescent transient device, so-called LIFT, for proximal sensing of chlorophyll fluorescence. And what it does, it sends a blue light. Here you see it being sent in the canopy of wheat. And it can receive many parameters, but two, the very well-known photosynthesis, the variable fluorescence divided by the maximal, and the other one is reoxidation efficiency. And these are both retrieved from one simple transient. They don't send a saturating flash, but they use multiple, in a very high frequency, small flashes of blue light. And because these flashes follow so quickly after each other, you also get a saturation of your photosystems. The saturation of the photosystems here is shown by the green light in fluorescence. You get a maximum, and this maximum can also be used to measure your variable fluorescence minus your maximal fluorescence. This approach I would like to show in this talk. I would give a short overview, not using it from a plane, but just a quick overview in different photosynthetic efficiency approaches in proximal sensing, covering a drought response in soybean, a drought response in durum wheat in Maricopa, a CO2 response, and a biotic response. So 4 examples with which I would like to finish my talk. Starting with the soybean in Embrapa, the experiment there. The experiment, well monitored there also the environment. Depending on the soil water content, they irrigated in this field with this irrigation bar, with at each level of the irrigation bar different levels of irrigation. You can see on the right side a nice overview. In the center there's the highest irrigation, and towards the side of each plot on both sides there's up to 5% irrigation only. Using this system where you have a nice control in a semi-natural environment during the dry season in Brazil, in Embrapa Cerrados, you can measure plants grown on the different soil water contents. And there we quantified by driving over here with the lift sensor, in this case in a black machine, on a pretty simple positioning system. And what you see there on the right upper side is that the photosynthetic efficiency decreased with the amount of water given. So only at 1% irrigation you had the lowest photosynthetic efficiencies. If you look at the picture here, it's rather obvious. It's also not what may a farmer say, oh, yeah, we can see that by our eye. The interesting thing is the stage before, but we didn't happen to be there, we were there last year, but this is an older slide, that you can see the photosynthetic efficiency also decreasing when there's no effect visible yet on the plants above ground. With that, I would like to continue a little bit bigger and also a little bit in the invisible parts. This is a large experiment in Maricopa where they grew a lot of durum wheat varieties, and also there we went with our lift sensor. Here on the left side, mounted on a bicycle platform from Jeff White. And Nicolas Senonadi, a PhD student in our lab, he went there and he measured those 252 genotypes from the starting point when they stopped irrigating. Also, this was in March this year, in the dry season of Arizona, no rain at all. But they choose there to stop the irrigation, and then we followed the development of drought stress among these 252 genotypes. And then we harvested. And here you see the results. Oh, that's vibrating. Oh, okay. Here you see the results over time. So it was started on 11 March. Photosynthetic efficiency was still pretty high. And as drought developed, so they stopped irrigation actually on the 11th of March. And then over a period of about 2 or 3 weeks, the photosynthetic efficiency decreased, similar to what we saw also and what we expect in Brazil. But what you also see on the right side, where if you have spatial distribution of all the different genotypes in the 2 replicates, above the dotted line is one, are the same genotypes as below it. You see the variation of all these genotypes in response to the drought. And with this data at the moment, we're doing the GWAS analysis with Roberto Tuberosa to find all this photosynthetic response in relation to these genes. That was an example of a large data set in durum wheat. And then we move to another data set, a little bit more future outlook, where we look at the response of photosynthetic response on the elevated CO2. For that, we moved to Germany, where we set up a breed phase installation, an installation to grow genotypes on the elevated CO2. It's set in a very rich soil area in Campus Klein-Altdorf of the University of Bonn. And we continue there with normal agriculture practices and combine that with non-invasive phenotyping. Here's how it looks from above. We have 3 phase rings of about 20 meters in diameter. And in each ring we have 42 plots to keep them just large enough that the breeders were happy to have a reliable yield of 1/2 by 3 meters. And this collection that we grew here last year was, this year actually, was a collection of historical collection, so released from 1963 to 2013 to the market. It was also an infrastructure set up by the German Plant Phenotyping Network, and is, or I must say was, accessible because we were rather successful in obtaining several projects from EPPN 2020 to do research in this facility. But do go to the EPPN 2020 sites because there are a lot of more facilities available to do your research. What we didn't have at that time yet, what's on the develop now, is an automatic positioning system. But as I said, it's not always the positioning system that determines the data. It was this: we used our bike with a little extension to measure over the plots on the elevated CO2. And we did that, or actually, I must say, Vikas on the right there, he mostly did that together with Andre Krause. And they measured at regular intervals. I was specifically interested in the early growth stages in wintertime, so the interaction of temperature and elevated CO2. And what you see here is days after sowing. So we are starting there around February, and we're measuring until about April. And you see that the reductionation efficiency on the y-axis is increasing over time. This is an increase with temperature, more or less, and the drops that you see there are also related to drops in temperature. And the other one, it's treatment versus control. The open symbols are the control and the closed symbols are CO2. To visualize that a little bit better, I choose to plot the relative response of CO2 versus control. And you see that the photosynthetic efficiency for the reoxidation efficiency actually over all the months consequently decreased on the elevated CO2. We can think about the mechanism behind there. You can think about the photoinhibition, the feedback mechanism, where it's cold and plants cannot get rid of their sugars and decrease their photosynthesis. That's actually what we could be measuring here. Unfortunately, we do not have the sugar content measurements or sucrose, hexose in it. But what you also see towards the end is this difference disappeared. You may wonder why. Well, luckily there I do have an answer. because that's where we stopped the CO2 for quite some weeks. We had some safety issues and technical improvements to do. So in April, end of April, we had to stop the CO2, and you see also quickly after the response of photosynthesis also stops. You see some variation among the different genotypes. Some genotypes that suddenly behave different, but that may be some errors in the field. So that was the response to CO2, and I promised one more data for examples of data combined with lift, and that was in a more biotic interaction where we look at SUGARBEET CERCOSPORA infection. Here we see a row of SUGARBEET plots in the south of Germany, and again we brought our lift sensor there and we measured over these plots. There was some natural inoculation, and they also actively inoculated different SUGARBEET varieties with different sensitivities over time. And when we look at the data over there, on the x-axis you see SUGARBEETs that have a low sensitivity to Cercospora, and resistance, high sensitivity, and a very high resistance Cercospora on the right side. And then you see two lines. The upper line, the SUGARBEETs that were not inoculated or were inoculated naturally. And you see that the ones with the lowest resistance have also the lowest photosynthetic efficiency. And interestingly, you see also that there's a response of the inoculation. When there's a higher pressure or more Cercospora, you see that overall the photosynthetic efficiency also dropped. So with this, we could also have a nice integrated approach or check of resilience or resistance of the sugarbird population. So that's the last example I would like to show you. And with that, I would like to thank you for your attention, and also thank the collaborators and all the people. Of course, this was not only my work, but a large group of researchers in IBG2, the Forschungszentrum Jülich, together with, and especially in the group of Shoot Dynamics. And with that, I would like to end my talk, and thank you for your attention.